All right, so this lecture and the next one will deal with the regulation of the blood pressure. And what we mean by regulation is those mechanisms that come together in order to maintain the constancy or the stability of the blood pressure, but also those factors that lead to chronic long-term changes in blood pressure. So today's lecture we will discuss what we are calling short-term regulation, and I'll tell you what we mean by that in a minute. And in the next lecture we will talk about long-term regulation. But before we talk about regulation of blood pressure, let us define which systemic arterial blood pressure we are talking about, because as you know, blood pressure has two components, a systolic component and the diastolic component. And they can differ in significantly, sometimes by 30 or 40 or 50 millimeters mercury. So when we discuss regulation, which blood pressure are we talking about? In order to be able to have a unified value for the blood pressure that is physiologically valid, we introduced the concept of the mean arterial pressure. And this we do to define a physiologically valid single value for the arterial pressure. Now, so how do we actually calculate the mean arterial pressure? The mean arterial pressure is the mean pressure experienced by the circulation. It is a calculated number. And because the diastolic blood pressure lasts twice as long as the systolic blood pressure, we multiply the diastolic blood pressure by 2, we add the systolic blood pressure and divide by 3. So the mean arterial pressure, therefore, is normally much closer to the diastolic blood pressure than to the systolic blood pressure, simply because diastole lasts twice as long as systole. Now, what is the importance of mean arterial pressure and why do we use it? We use it as a single blood pressure value that allows us to calculate resistances, which include the total peripheral vascular resistance as well as vascular resistance in specific organs. Now recall that mean arterial pressure, or pressure in general, is the product of cardiac output multiplied by peripheral vascular resistance. Since we can measure cardiac output, we can then divide mean arterial pressure by cardiac output and obtain a value for peripheral vascular resistance. We can also, if we measure renal blood flow, which we can do, obtain a value for renal vascular resistance by dividing the mean arterial pressure, which is experienced by the kidney as by any other organ, divided by renal blood flow, and therefore obtain a unique value for renal vascular resistance. So what we mean by short-term blood pressure regulation is the regulation of blood pressure in time spans which go from second to second regulation to hour to day. So systolic and diastolic blood pressure values vary from second to second, from minute to minute, and hour to hour, and during the day. And this type of variability, however, remains within usually a narrow stable range with occasional ups and downs throughout the day. And this type of variability, or the values of the blood pressure throughout these limited periods of time, can be measured clinically by wearable devices, whether they are a 24-hour blood pressure monitor or what we call a phenometer, which would actually measure blood pressure variability from beat to beat. But the most commonly used way of looking at the variability in blood pressure throughout the day is a 24-hour blood pressure measurements that the patient will wear a device that will measure the blood pressure throughout the day. And this gives us an idea of what so-called diurnal blood pressure profile is. And it's depicted in this curve here, where we can see, if we look at the systolic blood pressure in the upper curve, the diastolic blood pressure in the lower one, that starting if we look at from noon time and then going to 3 o'clock, 6 o'clock, 9 o'clock, and then the night time, how the blood pressure dips during the night and then goes up again during the day. But notice that throughout any particular hour or throughout the day, there is a lot of variability. There isn't a single value for the blood pressure, but all of them fall within a rather narrow acceptable range. So this is what we mean by a short-term variability of the blood pressure, and we will go into a bit more detail into the control of this short-term variability the rest of the lecture. But before we do that, let me just tell you what we will be talking about in the next lecture, which is what we systemic blood pressure. Let us focus a bit on what we mean by regulation. Regulation or maintenance of homeostasis or stability requires two major components. It requires a sensing system which detects or monitors the blood pressure from minute to minute, from second to second, from hour to hour, records these acute increases or falls, and then triggers a secondary response system to attempt to restore the blood pressure to its baseline value. So, therefore, there are two major pathways, therefore, for the regulation of the minute-to-minute or short-term regulation of blood pressure, which target either the cardiac output or the peripheral vascular resistance, since these are the two components which together determine the mean arterial pressure. Now, if you look at peripheral vascular resistance, this is principally regulated through neural pathways because they respond very rapidly within seconds and minutes. They affect the diameter of small blood vessels through vasoconstriction or vasodilation, and therefore immediately change the peripheral vascular resistance. Now, the sensors for the peripheral vascular resistance changes through the neural pathways are what we call high-pressure baroreceptors and chemoreceptors that are located in the carotid circulation, and we will discuss those in a minute. On the other hand, the regulation of cardiac output, which is the regulation of effective circulating volume, is detected in low-pressure systems, or what we call low-pressure baroreceptors, which are located in the atria and the vena cava. We will look at those at the end of the lecture. And when these detect changes in volume, either up or down, then they will regulate the effective arterial volume principally through altering the renal salt and water excretion rates, and therefore either retaining salt if the circulating volume is low, or excreting salt if the circulating volume is elevated. So if we look first at the regulation of peripheral vascular resistance through neural pathways, which respond, as we mentioned, very rapidly, and which proceeds through these high-pressure baroreceptors and chemoreceptors, let us examine how exactly resistance is varied when you change the radii or the diameters of very small blood vessels. So this becomes clear when you look at the formula for vascular resistance or for resistance through any tube, because resistance in a blood vessel is inversely proportional to the fourth power of the radius of that blood vessel, as you see here in this formula. And therefore, by changing the radius of very small vessels, such as arterioles, via rapid neurohumoral responses, you can vary peripheral vascular resistance quite profoundly up or down, and therefore mean arterial pressure can be regulated effectively. Now, where in real life are these vascular resistance vessels located? If you look at the curve on the left here, just ignore the one on the right, which shows the systemic circulation as you go from the left ventricle all the way to the vena cava, you'll see that the pressure drops most profoundly when you cross the arterioles and then the capillary network, but it is at the level of the arterioles that the pressure drops most profoundly, and therefore the arterioles are the main resistance vessels in the body, although there is some further reduction across the capillary network. So let us now look a little bit at these detection mechanisms, or where are the nerve endings for these systemic baroreceptors and mechanoreceptors that detect acute, small, or large changes in blood pressure, and how do they actually relay that to the central nervous system. So these detection centers are composed of specialized nerve endings. They record acute increases or falls in mean arterial pressure and relay these to the central nervous system via the vagal and the glossopharyngeal nerves. So the afferent ends of these pathways are located in the carotid bifurcation, the area known as the carotid sinus, as well as in the aortic arch. And then they carry the afferent pathways, or they send their signals to the central nervous system via the tractus basically to the tractus solitarius of the medulla through two major cranial nerves, the glossopharyngeal nerve and the vagus nerve. Now, the responses come out of the medulla, again through the vagus nerve, which goes to the heart, but also through the sympathetic network, or which causes sympathetic vasoconstriction or vasodilation fibers to the entire vascular system. Now, this slide shows you the anatomic locations of these nerve endings, and as you can see, you have in the carotid as well as in the aortic arch. So what happens if you cut off these baroreceptors? This slide demonstrates the result of that maneuver. In the upper curve is the normal blood pressure, and as you can see, it varies, but it varies within a narrow range. That's because the baroreceptors are keeping a tight control of these values. If it goes up too high, then they cause vasodilation. If it goes too low, they cause vasoconstriction. If you remove them, then what happens is what you see in the lower curve, a total chaotic behavior of the blood pressure, which is totally inconsistent with a proper functioning of the body and of homeostasis. So these baroreceptors are extremely important in keeping our blood pressure variable, but within a narrow range of variability. So in addition to the high pressure baroreceptors, there are other receptors which also can trigger neural responses, and these are the chemoreceptors, which are evolutionarily very primitive and old systems that are not very important in the day-to-day physiologic regulation of blood pressure in humans. They become important under conditions of profound stress, such as with severe brain hypoxemia. They start firing, but to our purposes, they really are not that important from a clinical point of view. And they are of two types, the peripheral and the central. And their locations are again in the aortic arch, the aortic bodies, and in the carotid bodies. They're sometimes called glomus cells. And their major stimulus is a low oxygen tension in the bloods, or low partial pressure of oxygen, which triggers firing from these glomus cells, again going through the glossopharyngeal and vagus nerves and causing the appropriate responses in the periphery. The central chemoreceptors are present in the medulla. Again, they get activated when the brain pH falls, and they lead to an increase in sympathetic discharge, become operational in brain ischemia. So these are fairly primitive mechanisms that are clinically not very relevant, but need to be mentioned. Okay, so we've discussed so far the regulation of peripheral vascular resistance through neural pathways, the baroreceptors and the chemoreceptors, most importantly the baroreceptors. So let's now turn to the other part of the regulation of mean arterial pressure, or the other determinant, which is cardiac output or volume. And as we mentioned, the regulation of cardiac output or effective volume is principally through the regulation of renal salt and water excretion. And the sensor mechanisms for that are what are called low pressure baroreceptors, which are nerve endings that are located in a segment of the circulation that is sensitive to intravascular volume. And that segment is the right atrium and the inferior and superior vena cava. So here is the anatomic location of these low pressure atrial baroreceptors, and they are of two types. There are the A fibers, which monitor heart rate, and there are the B fibers, which monitor atrial volume filling or effective circulating volume. So if they detect an increase in volume, then they will increase salt and water excretion, and if they detect a decrease in intravascular volume, then they will promote salt and water retention. Here is an example of what they do under conditions of volume expansion, such as occurs in day-to-day fluctuations in ECF volume. Because in the day-to-day challenges to intravascular volume are mostly in the direction of increased intravascular volume. We eat and drink all day long. We increase our intravascular volume because we eat salt and we drink water. And therefore, these receptors, which are located in areas that can detect these changes, will immediately trigger responses to help excrete the water and the salt. The first of these is the Bainbridge reflex, which is admittedly a neural one, but it increases heart rate, but it decreases renal vasoconstriction, so it increases renal blood flow, and therefore allows the kidney to increase its salt and water excretion. And then you have the effect that these nerve endings have on the central release of hormones, which are very much involved in water and salt excretion. The first is AVP, arginine vasopressin, or ADH, antidiuretic hormone. Same hormone, two names. When you suppress AVP, which tends to retain water, then you increase water excretion. And when you stimulate ANP, or atrial natriuretic peptide, then you increase renal salt and water excretion. So by manipulating these hormones and manipulating renal blood flow, these low pressure baroreceptors can deal with acute volume changes which tend to increase or decrease the blood pressure. So in summary, therefore, we have discussed two types of responses: those that are mediated by the baroreceptor and to some degree the chemoreceptor systems, and which result in acute neural changes that acutely increase or decrease peripheral vascular resistance. And then there are the low pressure baroreceptors, which regulate intravascular volume or cardiac output, which is the other component of mean arterial pressure. So the combination of these systems, we maintain our blood pressure on a day-to-day and hour-to-hour basis in a relatively stable values. Thank you.